4/100th of a millimeter, the diameter of a hair. It is a huge standard of measure for our subject, but it will help to orient us to the size of viruses. A red corpuscle of human blood is in this proportion to a section of a hair. Here is a comparison of the size of a red corpuscle, a typical bacillus, and two well-known viruses. The poliomyelitis virus, though not the smallest of the known viruses, measures 22 millionths of a millimeter. The electronic microscope makes it possible to see and to photograph viruses. Here is a series of photos taken by this method. The viruses have a variety of forms: globular, slightly elongated, or thread-like. Here is a red corpuscle which has absorbed influenza viruses. Viruses may cause disease in plants, animals, and human beings. They are complete parasites. In order to reproduce, they must attach themselves to a living cell. Here is a reproduction cycle followed by many of them. The virus penetrates the cells, it seems to disappear. It breaks into particles incapable of causing infection, and finally each particle grows in size and forms a complete virus. At 70 degrees below 0 centigrade, viruses will maintain their properties unaltered for a long period. A plastic holder contains slender glass vials in which are congealed a few drops of infected liquid. Research in this field offers great possibilities for the future of medicine, both in the isolation of still unknown viruses and in the development of more efficient methods of control which give a stronger guarantee for the use of vaccines. Viruses can be made to reproduce in receptive animals such as ferrets and mice. Monkeys are used especially for research upon poliomyelitis viruses. Fertile eggs are one of the best cultures for viruses because they offer a sealed, sterile place for growth. The cells which line the egg cavity and form the embryo are highly susceptible to a great number of viruses. The position and vitality of the embryo are ascertained by examining the egg under ultraviolet rays. In order to withdraw the virus which has been reproduced, the egg is opened at the air chamber end. A few drops of a suspension of red corpuscles on a glass slide is an indirect method of revealing the presence of influenza virus, for example. In fact, this virus has the faculty of coagulating red corpuscles so that when this phenomenon is present, the infection is evident. Especially interesting for the study of these phenomena are cultures in test tubes of cells which are the sphere of infection for viruses. Here is a view of a culture of cells which demonstrates graphically the elements of their composition. Externally, the cell is confined by a membrane. The interior is made up of cytoplasm, the microsome, and the mitochondria, the nucleus, and the nucleolus. Here is an actual single cell. In order to cultivate live cells on a culture dish, a fragment of tissue must be obtained in a sterile manner and subjected to a process which causes the cells to separate from each other. A special substance called trypsina aids in this action of separation. The mechanical action of special agitators gradually reduces the number of fragments and induces the complete separation of the cells. Finally, centrifugal force exerted upon the liquid causes it to deposit the detached cells in a test tube ready for culture. Today it is possible to maintain in continual culture groups of animal and human cells which live and reproduce in special receptacles completely isolated from the original tissue. Here are living human cells obtained by culture. These are from the liver and have already been living some time detached from the original tissue. These, which are also alive, come from human kidneys. These operations appear extremely simple, but are really almost miraculous. By means of them, science has been able to prolong the life of a tiny part of our organisms, the cells. This victory of man over matter has made possible one of the most useful methods for the direct study of human pathology. Thus, in many cases, medicine is able to isolate diseases and study remedial drugs under ideal conditions by substituting living human cells for human beings in experimentation. Here are virus-infected cells as seen under the microscope. They are pictures of a frightful reality, which, however, do not reveal to the layman the process of destruction which has extinguished life. To the experts, they represent only a single moment in the process of infection. A new experiment might prove useful for observation and study, the use of the motion picture. The Istituto Nazionale Luce and the Institute of Health determined to make the experiment. The camera is being mounted for microscopic filming. A chamber with a constant temperature allows the cultures to be maintained at 37 degrees centigrade in order not to alter the cells. Meanwhile, physicians have studied and selected the materials and decided upon the methods best adapted to filming. Months of preparation have passed. The long-awaited moment has arrived. The parts of the filming chamber are taken from the ultraviolet ray sterilizer. The physician is ready to furnish the material to be filmed. The slide which will hold the cells is infected with the virus. It is a very delicate operation. Here there is direct contact with agents of the most terrible diseases. The slide is moved into place. Final preparations are made. In a few moments, we shall know if the experiment will have positive results. Ready. The camera exposes one frame of film every 15 seconds. A fraction of a millimeter separates the microscopic lens from the infected slide. Here are the first dramatic film shots. A group of cells. The virus has already attacked one which has contracted spasmodically. The phenomenon is called boiling. A colony of cells after a few hours of infection by the virus which attacks the respiratory system and the eyes. At the left, there is a cell with evident signs of the phenomenon. The central part contracts and becomes grainy. The exterior forms elongation. It seems to be using them in a desperate effort to grasp the slide of the nearby cells. However, its doom is sealed. Another view of cells infected by the same virus. The cells contract, showing the intercellular spaces very clearly. The same phenomenon takes place at the lower right. Very tiny intercellular bridges are evident. Note the cell above and to the left. During a contraction, a prolongation is projected and immediately retracted. Now there is another small one which quickly disappears. Reduced to an amorphous mass, the cell is now passively pulled about by the force generated by the contractions and movements of the other cells. The death throes which the motion picture camera has permitted us to follow in their entirety in a few minutes actually lasted about 15 hours. A cell colony which has been infected by the virus of poliomyelitis. Notice at that the tragic phenomena of the reproduction of the virus and the death of the cells. In many other cells, signs of suffering appear around the entire periphery and in toward the center. Cellular contractions accompany the boiling phenomena. Long protuberances agitate freely, the obvious expression of the biological cataclysm taking place before our eyes. One by one, the cells are inexorably doomed. The actual duration of the phenomenon is 4 hours. We have seen the first motion pictures of human cell colonies of unicellular derivation. In its unceasing fight against disease, medicine has achieved a new means of